Yeah, I'm kind of.
I want to get him a new stuff on, but now let me now, what Melissa did was a big R, a big B, small r. You know what she did wrong, right?
Yeah, I heard.
On the fish. She knew that red was recessive. What letter should she use? Not small r.
Small b.
Small b. All the small b says that it's the other way. The big B says blue is dominant. And then when you put down just, I know what you're doing. Many people can do that. But you must stick with the same letter every time, because otherwise, if you have 4 or 5 letters, that's going to drive you nuts. So anytime you see a B, if it's a big B, it's blue. If it's a small b, remember, it's red. Okay? So that's the first thing you got to watch out for. Is get the same letter. I wouldn't use a small w here, no, he's white. I want to use a small p, meaning the other condition. Now, these are alleles. There's 2 alleles here, 2 alleles there. If they're the same, they're homozygous dominant and homozygous recessive. If there's one each, it's just called heterozygous. It will be dominant, but it's understood. There's your 3 combos. There's another way you can do it. And you always write the big one first. That's just why you always capitalize the first letter of a sentence. You just do it. Okay? The letters represent the type of genes. We call it genotype. And then when I say purple versus white, that's the phenotype, physical, how it looks. And like I said before, if they're the same, the word is homozygous, but you must tack on whether it's homozygous dominant or homozygous recessive. If they're opposite, like these 2 are, we just say hetero for different zygous. Okay. The first law says that when this plant makes his pollen, one pea goes into one pollen, the other pea, we split them, we segregate them. And this is the other plant, and this might in the flower area, but one pea goes into an egg. So when you segregate, you actually split the alleles. Now, in the adult, they're always in pairs, right? So if these 2 were to find each other and make a plant like this, big P, small b is still going to be your purple plant. I didn't say big P, small w now, Melissa.
I know.
Okay. Big P, small p. And you got to remember that the hard thing is keeping track of what the small p is. Okay? Now, when these cross each other, the only combination is going to be big and small. There's no other way you can do it. They're all going to be big and small. Every one of them. You're going to lose your white flowers. You're going to lose that red fish. Because the big letter always kills the small letter. So no matter how you played it, if these 2 got together, or those 2 got together, it doesn't matter how you do it. They're always going to be big and small, heterozygous. Now, what I like to do is make my Punnett square. And you see right there? These 2 are on this side. You see those 2? They're across the top. Now combine them. I pretend that this pollen found this egg. That's the big and small, right? I said, well, I pretend this small. Well, they're all going to be the same. They're all going to be exactly the same. There's no way to get a white flower, is there? Because that big pea always going to kill. And now, you kind of wonder, where'd Mama go? All the white flowers. Well, Mama's still there, but she's hidden by the dominant gene. Now, if you let these 2 cross, then there's a chance this one can find that one, and you will get your white flowers back in the 2nd generation. Okay? Now look carefully now. This is heterozygous. Now I'm going to pretend that 2 of these got together. These 2 are 2 of those. Now I want to let these 2 pollinate. This goes into one pollen, that to another pollen.
That'll be your F2?
This will be your F2s. This is your grandchildren. Okay. So when you do the segregation, on this side of the Punnett square, you'll have the big P, small p. While on top, you have the same thing again. Because both parents are identical. Okay? So there's your segregation. Same both times. So when you do your Punnett square, that's just how I do it. When you do your Punnett square, you're going to have, oh, I changed up on you. Let me go back. I wasn't going to do this anymore because I didn't want you to learn the same thing over and over again. And I'm going to work now with them tall plants. And since tall is the dominant, I'm using the big T. So short is the recessive. I use small t, not the X. Small t. Now I got to remember what the small t stands for. I just got to remember it stands for small. All right. So I got this purebred tall. And I got purebred short. So he's going to be big T, big T. This will be what? Small t, small t. Now I cross them. Every time you see a big T hit a small t, who's going to win? He's going to be tall. So, if the tall P's will look like this. We said, and I'll put, you can put the big T's there, it doesn't matter where you put them at, but I always put them on the side over here. So, the two T's, I'm going to put one each right here. The two smalls, I'll put one each right there. And I'll come back. See, I know by the rules that when you cross heterozygous individuals, you're going to have this 3-to-1 ratio. 3 going to be tall, 1's going to be short. Now, if you look carefully, you'll see how it works. These two come together, and everything you see there, these are all tall. Because this is homozygous cross homozygous. Now, when you cross two of these, now you have a chance that two small t's get together and making you a short plant. But when you cross this, there is no way to get a short plant because he will always cancel it. Always cancel it. But this is possible. Now, if this over here was big T, small t, they'd both be tall plants with it. If he were a big T, small t, and you crossed them, could you ever get any small plants? Still going to make them all tall, right? But the genotype be different. I want to have two which are homozygous because I got this can happen to a big T. I don't want to confuse y'all because I'll start to see things abstractly. Let's combine them.
You get one that's short, right?
Well, if you got two T's over here, you'll never get a short plant. If one parent's got two big ones, you'll never get a short plant.
I thought you said if you make a tall one and a short one on both sides.
I make him a tall short. You still won't get a tall plant because he's going to always kill it.
Mm-hmm.
All right. Now, if that's a small T right there, you shouldn't do it because there's a chance that this T could find that. It's going to be 50-50 then. Okay? We're going to cross, we're going to let two of these cross. We're going to let two of these cross. Okay, first off, they're all heterozygous, right? All heterozygous. The genotype, if you're heterozygous, you're going to be the dominant trait. You're going to be tall. That's your genotype. One each. The phenotype, you're tall. Now that's the F1 generation. That's the cross between a purebred tall and a purebred short. They're all going to be tall, but none are going to be purebred. Okay? Now, if I let two of these cross to get my F2 generation, my second generation. Now, we'll do it before I get to my homework. Let's do it. If I cross two of these, it's going to be big T, small t. Big T, small t, right? Now, if this big T joined that big T, what do you have? What kind of plant? How do you look? Tall. Homozygous tall, heterozygous tall. Now, now, if this small t finds this big T, it's going to be big, small, right? How will it look? Homo or hetero?
Hetero.
Hetero. Now back here. If this big T finds this little T over here, then you got big, small. How's it gonna look? Tall. Homo, hetero.
Hetero.
Now if this small T finds that small T,
Homo.
you got your short. And to be short, what must his genotype be? He must be homozygous. Because if he's heterozygous, he can't be short. And when you cross hetero and hetero, if you look, you got 1, 2, 3 with a tall. You got 1 short, 3, 1. But if you look at the genotype, I got homozygous tall, homozygous short, and 2 heterozygous. So phenotypically, it's a 3-to-1 ratio. If you had 100 plants of that combination, 75 you expect to be tall, 25 you expect to be short. It might be 73 versus 27. It's still 3-to-1 roughly speaking. Now, I don't want to discuss this because some folks still working on it. Now, this is kind of new stuff. And when I saw I'm about to get into a double trait. It's not bad crossing tall plants and short plants, or crossing blue fish with red fish.
Let me ask you a question before I confuse myself, because I think I just did that wrong. When you did that first generation, that small one and the small t, big T and the small t, it's not a 50-50 of tall and short?
If that first generation, if one of those plants is homozygous dominant, you won't get anything but all tall. Because there's no way to get rid of, every pair you have, you're going to have a dominant gene. If both of those plants are purebred, and one's purebred tall, and one's purebred short, all the tall can give you is the big allele. You're always going to match the other allele.
That's the F2 then.
Oh, the F2s? Okay, the F2s now.
The tall T and that little T.
F2 with the heterozygous, then you always got a chance that one small T can find the other small T and get you a short plant.
That's a 50-50 then.
Uh-huh.
Where is the other one coming from?
Let me show you. Let's take an, or there's like a single trait, color of the fur, how tall the plant is, what color the flower is. But what if you're going to start crossing a tall plant with white flowers? What's a short plant with purple flowers? What kind of plants you going to get? That's a dihybrid. That's 2. And Lord, you try to do a three hybrid, where you get I want a tall plant with purple flowers and the flowers up and down the stem. I'm going to cross that with another plant. This is a short plant with white flowers and the flowers at the end of the stem. What will I get? That will be a tremendously big Punnett square. And a trihybrid cross is fine to do. But if you got, if all those folks in all those things are heterozygous pairs, oh Lord, you got to put in a square. You got to glue paper together, put it all on. All right. Well, let's talk about it in a minute. Then we'll have it. Okay. Let me make it bigger. Okay. Actually, to me, a monohybrid cross is boring. It really is. I mean, you can do it in your sleep after a while. I like the bigger crosses because I get to really stretch my brain a little bit. But let's talk about it. So a mono, what's mono mean? It means one. Hybrid means trait. Hybrid means trait. So if you just look at one trait, say it's flower color, that's monohybrid. Let's say it's the color of the fish, blue fish, red fish. That's just one trait. Question is, how would you set up and do a Punnett square if you're crossing 2 traits? Like a plant with purple flowers up and down the stem, with one with white flowers on the end of the stem. Those are 2 traits. One trait's flower color, other one is flower position. That's the dihybrid. Now, I want to show you again what's the boss. Now, if you look at these things, and of course you don't have colors, let's take a picture of the screen. I don't know where you can find this at. It might be in your book somewhere, because Mendel's, these traits are very, very famous. If you look, what we're talking about, the flower color is always these two colors. And this column is always a dominant trait. This is always recessive. So there's your dominant, there's your recessive. If you look at them all, do you see any words start the same letter? It made it nice. So for seed shape, we had big S, small s being wrinkled. For seed color, big Y, small y being green. For the color, big P small p being white. For the shape of the pod, inflated like fat, that's a big I, small I. And that's your problem. Big I, small I look alike. You need a line under small I then. Well, these S's look alike too, unless you use a cursive. I'd put a line under them. The color, big G, small g. Those G's look different. No, big G, small g. You can tell those don't need a line with those. Position is the big A versus small a. Big A being up and down the stem, small a being the very tip. And think about it. Between this, say you got a tall plant, between this and this, who could have more flowers? Tall, up and down the stem, or a tip of the stem? Who could have more flowers? Up and down. Would that be advantageous? Yeah, you got more you can pollinate. Of course, now if you're a short plant, it'd be more advantageous to have at the end, out here. And I got room for 3, but I put 3 right there. So these traits work out to be sometimes pretty good for it. But tall versus short, and we don't ever use the small s, don't use a small t for this one. I always use the same letter, lowercase. Now, I think we're going to do the purple flower up and down. This I'm using this trait and this trait. There's up and down, and there's at the tip, terminal. All right. And I made this thing up. I took and I used my paintbrush and I did all kinds of stuff. I'm going to pretend that this, okay, this, they're up and down as you see there, and they're all purple. We don't know if the sucker is heterozygous or homozygous. Now, did I ever study their pure breeding? Oh, they're pure breeding. Pure, okay, good. See what it says pure breeding right there? It tells me big P, big P. Up and down the trait. Up and down, big A, big A. Right there. I put them side by side. Purple flowers. Ooh.
Yeah, blue.
Ooh, that ain't right, is it? What's wrong with this one here? What's wrong with... I'm wrong.
This would be both, all 4 letters counted?
Yeah. It should be both. That's what I'm trying to teach y'all something. Sometimes I do them wrong on purpose, see if you can catch me. I think I was messed up that way. All right. So dominant. Now they're pure breeding. They're pure breeding. Therefore, they're going to be 2 big P's. They're also pure breeding for up and down. That's going to be 2 big A's. So this is one plant right there. That's one plant right there. Okay. So this is my genotype for 2 traits. Right there. Now I showed this again to you. Now look at this thing here. Oh, you know what? I wanted this. Let me see. Yeah, I wanted this to be a small a. I think I know where I was going. I want that to be a small a. This is not a pure breeding plant, because that's heterozygous.
It is a pure breed.
Wait, that's wrong. This one here. I wanted this to be a big A. I want it to be heterozygous. I don't want this to be a pure breeding plant. This one just happens to be purple flowers axial. But the genotype, I'm telling you, is homozygous, dominant, and heterozygous. We're going to cross this bad boy in a minute. I'm going to tell you this plant here, I'm going to tell you right now. The dominant flower is purple. That's why I must use small p's down here, right? Because purple can't be a big P, can it? If it's a big P, what color would it be? Purple. So I can't use these big P's here. Now, axial, when an axial is dominant, so if you have this right there, he's axial. That ain't axial though. You gotta take and make that that. And what you see right here, I'm just telling you, purple is dominant, that's why I got two P's. Axial is dominant, but this is terminal. This is terminal. Axial will be up and down the stem. So to be this way, he must look like this. Follow? Let's go back.
Shouldn't that say white?
Huh?
Shouldn't that say for the first one, the flower covered trait?
No, I'm making a statement. Dominant is purple.
Okay.
But he's a white one. Therefore, to be a white, he can't have big P's.
Yeah, too small.
I'm just information.
Okay.
Okay. Now, are there other genotypes that can make this? What else can make that?
P little b, a, capital, and little a.
Yeah. What else can make it? How about two big A's and a heterozygous P's? So there's more than one combination, right? I mean, if this is homozygous, would that be that way? Homozygous, heterozygous. Yeah. This is heterozygous, homozygous. Yeah. How about hetero? I got 4 gene types to have the same look. But I'm just saying right now. Oh. Since this is pure breeding, oh Lord, that can't be. That's got to be A. Right there is what I didn't read. Since he's pure breeding, what must he be? If he's pure breeding, everything's homozygous. So, now, let's go to the next one. Now, okay, this is pure breeding, that's going to be okay. Correct? Because if you're white, you can't have no big P, can you? And if you're at the very tip like these are, you can't be a big A. Because a big A has them up and down.
What does the other one look like again? Go back to the other picture. The
What are you asking me?
Oh, okay, that's what it looks like. Never mind.
Let's do something. And I don't want to mess people up. What did we say the other phenotypes were? We could have been what? Could we have been this? And look like that? All right, now on the next one we had here, and I did say, again, pure breeding. So what we have is all caps versus all lowercase. Now if you look at this, I'm going to take, and I'm going to change this. I don't know what I was thinking when I did this slide. I sure don't like what I had done. All right. Let me show you what I'm trying to take. But I better say that. Okay. I'm taking this plant, which is showing all the dominant traits, purple flowers, up and down axial. And I'm going to cross it with this plant, which is also purebred, showing all the recessive traits, white flowers at the tip. And that's why this one must be big, 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 big. This must be small, small, small. Now, if you do your segregation, always got one big P and one big A. I have 4 pollen. They all look alike. So I say it 4 times. That's going to be the only kind of pollen that plant can make. You follow me? I can do it 4 times, and why do that? And on this plant, that's the only kind of egg that that plant can produce. There's no other combinations. Now what happens if this pollen from this plant finds this egg? Well, that's right there is going to be your answer. What does that plant look like? Does it look like the white one or the purple one?
Purple.
Exactly. But is he the same genetically? He's just the same phenotypically. And I tell you right now, this thing right here, I put it right there, like that, and I draw a few more of them just to make it interesting. They all are going to look just like the other plant did. See what I'm doing? Question that Mendel had was, where are my white flowers at? And where's the tip arrangement at? And he said, ooh. So, everyone you have, they all look like, if that's daddy, I mean, they look like the purple plant. But just because they look like that plant, they are not the same genotypically. And when you cross 2 of these, you will then get all combinations. You'll find some purple on the top, some white along the sides, and I'll show you all kinds of things. That's when you get your real pretty roses. You get all these combinations from just 2 individuals. So right now we're saying that these are called the F2s. Matter of fact, let me see. This is the F2 generation. It's called the first spheler. So right there, all these are the F2s. No, F1s. That's the first of these.
And after that, you get your weights back.
Yeah. If I cross these, my F2s, my whites come back. But my F1s are totally dominated by the dominant allele. The recessive allele is hidden there, but it can't show us it because it's being covered by the capital letter. Now, okay, so this is the F1s. These are the initial parents. These are parents F1. Now, if we let 2 of these cross, I think I'm going to do that, I hope. Okay, now, and I already answered it here, but my question is, for the genotype you see there, which of those would be that genotype, A, B, C, or D? Which one would be the genotype?
D.
D. Delta would be. Because what's the dominant color flower?
Purple.
Well, the big piece is. And what's the dominant position?
Axial.
Axial, because the other one, tip, is terminal. So this would give you this. Now, the position right here would call for what letters over here? Both what kind? No, that'd be along, this is axial here. This is axial. For this to be there, it's going to be 2 small letters. No, I don't know, not the color. I mean where you find the flowers. If you find them up and down the axis of the plant, we call them axial. If they're on the very tip of the stem, we call them terminal. This is dominant, so these white flowers would be small p, small p. But this could be big, small, or big, big, for that matter. If it were small a, would it look like that? Small a is recessive. Where do you find recessive trait? At the tips. Up and down is dominant. The term is axial. We use an A for that. If you're up and down the stem, you're the cap. Now, if you're at the tip of the stem, you're not axial. I can't use big A for you anymore. I must use small a for you because you're on the tip of the stem. So, I did all this not knowing that this slide was coming, and some of y'all still missed it. All right, now. We're now going to, and you see that slide right there? Let me crank the show up again because other errors are driving me nuts. Now I want to cross two of these plants because they all are this one. You see it right there? I'm going to cross two of those. Now there they are. Hetero, hetero. You see it? Hetero, hetero. I'm going to cross two heteros. Now, this is one of the plants. But they're both alike, right? So whatever he can make, the other can too. So now I'm going to do is this. Since when I type it, I can tell the difference in my letters, I'm not underlining. But it's possible that this P and this A enter one of your pollens. It's also possible for that same P, but the other A enter. We're separated now. But I could go this way, where I have small p, big A. And I could put the two smalls together, right there. All I'm doing is this. If there are 4 letters in the cell to start with, how many must be in the gametes? 2. Now, I ain't going to have 2 P's in there, am I? I got to have 1 letter of each trait. Well, I just start off there. Well, let's let this big P and that big A that could be one combination. But I could have combined this P with this letter, and I said, well, next time let's go this way. And I did that. Well, I got small P's. So I thought, well, how about these two get together? Now, I don't put the A in front. I always keep that P in front now. I know the A is a big A, but it's a second trait. Don't mix those traits up. So, again, you do it this way. Well, that's another way you can segregate them. I can put a small P with the big A. And if your P's come first, they always come first. But now I got 2 P's in there. The big P comes before the small P does. And then over here, there's one way you can combine them, and that's your 2 small letters. Now, then we talk about what happens if this one joins this one and that kind of stuff. And that's my. Now, here I'm showing. And okay, so this is the F1s. This is the F1, one of the plants. And that's how you could possibly get them combined up. Right here, I got to change this word. This is F. I'm going to make the F2, but I'm going to do it by combining 2 of the F1s. I'm going to make the F now. If I cross 2 of those F1s, they both look alike, and I'm changing colors so you can keep track of them. Okay. That's not right. That's not right. I got to go back and get one like this. Now, tip up and down. Well, that ain't right. I got to put this one on. Yeah. Okay, now, they are purple, heterozygous. And they're up and down the stem, heterozygous. So this one, I'm crossing 2 of those. I'm trying to show 2 of them getting crossed. There. I want this plant to pollinate this plant. They are both identical, are they not? Both phenotypically and genetically. Okay? Now let's do the segregation. I told you earlier, big P, big A, they might go together. Big P, small a, they might go together. How about small p, big A? That could go together. And the two small, that's all the combinations and no more. And this one does the same thing. Now, I'm going to have these four down one side of my Punnett square, and these four across the top of it. Going to look like that. Let me go ahead and run the show again. That's now. Now I'll put it back together for you because I'm telling you that this P is this P. And this P is that one. So these two come together and make a difference. And these two now. If you look at this plant right here, which of these? A, B, C, or D is that plant. A, B, C, or D is that plant.
B.
It'd be B. Because they're purple and they're up and down. Which is this plant?
B.
It look like that too? How would he look?
B.
Like that too? How would he look?
B.
Are these really the same genotypes?
Mm-hmm.
No.
These two are the same, tell me?
No.
What's the same about it? Phenotype. 4 different gene looks give you the same appearance. Now, you look at this one right here. Who does he look like? A, B, C, or D? B again. Who does he look like?
A.
Like this one. Because they're purple and they're not up and down. Right? Who does he look like?
B.
B. Who does he look like?
B.
B. Who does he look like? B. Who does he look like?
B.
B. Who does he look like? Uh-oh.
D.
This one. Because they're white and they're up and down. Who does he look like?
D.
White and up and down. Who does he look like?
D.
This one. Purple, up and down. Who's he look like?
A.
Purple at the top. Who's he look like?
D.
D.
Wait, which letter say?
D.
D, yeah. White, up and down. And this one here?
C.
C.
How many would you have that is going to be white and at the tip and all? There are 16 kinds. How many look like this one? Just one. Just one. How many are purple up and down?
Oh,
Purple up and down.
I want to count.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10. How many are white up and down?
Can you put little
1,
numbers?
2, 3. How many are white terminal?
1.
Only one is. How many are truly, totally homozygous?
1.
2.
2. Of those 2, how many are totally homozygous dominant?
1.
Homozygous recessive.
1.
How many of those have a homozygous dominant genotype for the flowers? How many are homozygous dominant for the flower only? Just flower.
1, 2, 3, 4.
What are you looking for?
Big P, big P.
Big P, big P.
4.
4. How many are that way for the axial?
1, 2,
Homozygous dominant axial.
4, 5, 6, 3, 4, 5.
See how you work it all out? Could you imagine doing that without the Punnett square? Just saying, you couldn't do it here, could you?
No.
I don't think I could do it there. But when you give me that Punnett square, I can tell you all about that stuff. And my question is, how many would look this way? Just look this way. How many would look like this? Now to look like that, what must you be? At least one big P. At least one big P, and what?
Too small.
And total small. How many have at least one big P and all small? One. Oh, all small?
Just one.
Two.
There's one.
And then
What's another one?
second above it.
Two. These two would look like this one. How many look like that? At least one each, right? Would you say a lot? How many look? Well, if you count them, I want just one. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Like this one. And to be this way, that won't do it. That would do it. Only one. This is only this one. And to look this way, I would need to have all small p's, at least one big A, right? All small p's, one big A. These three would look like this. Can you imagine doing a third trait? Where you had another letter here? To make that thing. I couldn't put it on the screen, it'd be so big. Alright, so now you look at this. When you got done, how many would you have that were pure breeding for the dominant expression? Pure breeding dominant.
One.
Just that one right there. How many pure breeding for the recessive? Just that one right there. Are these pure breeding? If you got a small p there, what may happen down the line? You might get another small p. That's not pure breeding. This bad boy here, oh now, this one, if you cross this one with any of these, let's get that right back, you know why, don't you? They're all capitals. This is your dominant trait. That's the one that's worth a lot of money. And that's worth more. So what's harder to keep? The dominant trait or the recessive trait?
Recessive.
It's hard to keep it. Those red fish cost more than the blue ones do. You know why? Hard to keep them. When you let them go, and next thing you know, all your fish look what color?
Blue.
They're blue, they're a dime a dozen. To get a red fish, though, you must keep that sucker separated. Because if he mates with the blue fish, you're going to have some more red. I tell you what, if you have a red fish and you mate him with a blue heterozygous and you get maybe 2 more red fish, you know you have 3, which are high dollar fish. But if your fish was purely dominant, you'll never increase your stock until you start letting the FO2s get together and cross.
Is that an actual recessive trait in the betta fish red?
Mm-hmm.
So there's only one recessive expression, that's the little P.
Yeah. Well, any little letter is recessive, but a pair means you're totally homozygous. No, if you're small p's, you're not purple. If you're small p's, you're white. 1 big P makes you purple. 2 big P's make you no more purple. You just same purple you were. Now you get the roses now. Now roses, that could be a purple rose. That could be a white rose, and that could be a rose with purple stripes through white. Roses get a blending effect. So Mendel was lucky he didn't do roses. P's are absolute one or the other way. And that's what they begin to be called Mendelian traits. They're either or. But you see how many kind of roses there are. I mean, they're roses, and all they do is crossbreed them. Because all those traits are blending traits. And it produces a great variety of beautiful roses, but you don't have anything like this. All right? phenotypically, these parent plants are all purple, and I'm not gonna make you go through the gymnastics. You can probably do it now. I'm just saying, how many look like the parent plants? In other words, how many are purple axial? A whole lot of them are, as you know. And genotypically, how many of them? I'm looking for this one. He's like the mama and daddy. So is that one. So is that one. So is that one. There are not many who are the same gene type as mom and dad. There are a whole lot of them look like mom and dad. But there's only a few that actually are identical to mom and dad. All right. For a human being, oh, time to stop it. Y'all so polite, you should give me a cutoff sign. I'm gonna show some human traits, but I thought were pretty cool. So we'll come in tomorrow, and I don't want to revisit slide 35 again. I'll start with human ear and nose. So I'm making notes of myself. Tomorrow I'll start on slide 136. And I'll put on the, I got the lab almost like I want it. It's going to be clams. We're going to dissect the clam tomorrow. I'll bring a screwdriver to help pry the sucker apart so you can get there and do the cut. Some of you folks like oysters might decide you don't like them anymore when you see what all going on inside. I don't like them anyway, so I don't mind dissecting the bad boy. But we're doing that tomorrow. We're going to dissect the oyster. I've still got to proofread to make sure that all the these and theirs are done right. And then be looking forward to it here. All right?